Hi everybody, my name is Jocelyn, and today I will be talking to you about a method that scientists have found to possibly convert every type of blood into universal O-type blood. But first, let's cover some basics about blood. There are four main types of blood: A, B, AB, and O. So what's so special about O-type blood? Well, it's a universal blood donor. What does this mean, you may ask? This means that O-type blood can be donated to not only people with the same blood type, but also people with type A, type AB, or type B. This is because O-type blood lacks a marker called antigens. Blood cells have sugars attached to them called antigens, which serve as a form of identification. A blood type, for example, has A antigens, and AB blood types have both A and B antigens, except O blood cells, which have no antigens attached to them. These antigens are what are used by the body to identify if the blood cells match the body's type. Antibodies will use the antigens to determine if it's compatible. If they are not compatible, the antibodies will come in and attack them. So this means that O-type blood can go unnoticed. So how can we actually convert blood types into O-type blood? The answer is actually pretty simple. It all comes from a bacteria found inside the human gut. You know, right? Who would have known? Well, these findings were presented at the most recent American Chemical Society meeting in Boston by Professor Stephen Withers from the University of British Columbia. How was this accomplished? It was actually accomplished using metagenomics. What is metagenomics? Well, metagenomics is a branch of bioinformatics that studies uncultured microorganisms in the context of their genetic characteristics that exist as populations. This is according to ScienceDirect.com. They first started by sampling microorganisms in the human feces against sugars that resemble the antigens on red blood cells. One specific enzyme caught their attention. This enzyme was found to be used by a bacteria in the human gut that is able to remove sugars from glycosylated proteins, which are also known as mucins along the human gut wall. Some of these sugars resemble the A and the B antigens and serve as a food source for this specific bacteria. Scientists were able to take the specific enzyme the bacteria uses to cut the sugars and found that these enzymes were 30 times more effective at removing A and B antigens from red blood cells than previous ones. Withers is now working with the Center for Blood Research to further validate these results and conduct more testing. This is a huge step forward because now scientists will be able to convert all types of blood into O-type blood, which means there will be more blood readily available to donate to people who are in need.